Welcome to our Genomics in 2021 and Beyond webinar. We've just been waiting for a few of our attendees to join us today. So I'm going to hand over to Ange in a moment to kick off our webinar. Firstly, I'd just like to introduce myself. I'm the course advisor for this programme. If you have any inquiries about applying to this course after the webinar, you can get in touch with me via studyonline@manchester.ac.uk. So I'll put that in the chat box and I'll be answering questions throughout the webinar. But over to Ange. Thank you for joining us.
Thanks, Jess. Welcome everyone to this webinar. We're really delighted to welcome you along. In the UK here, it's snowing at the moment. I don't know what it's like wherever you are, but delighted to certainly be inside in the warmth at the moment. So we're going to be discussing genomics and beyond in 2021 in this webinar, and we want to wrap up by obviously introducing a little bit around our course that we do in clinical bioinformatics and genomics as well. But first of all, let me introduce myself properly. So I'm Dr Ang Davies. I'm a senior lecturer in clinical bioinformatics and genomics at the University of Manchester. And I also lead the programme of clinical bioinformatics. It's a fully online PG certificate that we've been doing now for the last couple of years. So let me introduce my colleagues, Dr Jan Taylor and Professor Andy Brass. Jan.
Hi everyone, my name is Jan Taylor and I am the lead bioinformatics scientist for the Northwest GLH. So I'll explain a bit about what GLHs are in a bit.
Thanks, Jan, that's great. Andy?
Hi, I'm Andy Brass, Professor of Bioinformatics at the University of Manchester. So I think I've now been involved in teaching bioinformatics at postgraduate level for over 30 years. And it's just been really exciting to see a transition from a discipline which was very new to now, over the last 20, 30 years, something that's making such a real difference to real patient lives.
Thanks, Andy. Yeah, it's been quite a journey in the last few years. I think we started training clinical bioinformaticians back in 2013, I think it was. So me and you leading the course back then, and yeah, things have changed an awful lot. So Jan, you've been leading bioinformatics in the Northwest. You've sort of just began a new role in the Northwest over in Manchester. Yep. Do you want to give us a bit of an overview of how the new Genomics Laboratory Hubs have been set up in the UK and a bit of background around genomics in the UK?
Yep. Well, the redesignation of genetic laboratories has come really as one of the outcomes of the 100K Genomes Research Project, which was a government initiative, a 5-year programme of research to sequence the whole genomes of healthy and diseased people. So they had people with inherited diseases, healthy people, people with infectious diseases, and also people with cancer. Quite a lot of different types of cancer, and their aim was to sequence 100,000 genomes in 5 years and have a really good research data set to use in the future. But the legacy of that brought up the fact that there are a lot of differences around England in the way that the genetic services were being delivered throughout the different laboratories. So there was a range of laboratories set up in hospitals that grew out of particular specialities, and so that meant that tests offered in one hospital or one region were not necessarily going to be offered or available for people in another region. So this brought up the fact that there wasn't even access to genetic testing around England, and so from the 25-26 sort of specialised genetic laboratories, England has been remapped into 7 genomic hubs. So they're led by 1 or 2 centres in those regions. So there's 7. There's the Northwest, the Northeast, there's the East Midlands, there's 2 in London, Southwest, and sort of the middle of the country through Birmingham and Salisbury. And so in addition to redividing up the country in terms of laboratories, they've also developed a test directory, which is a national test directory. So that's split into 2 parts. There's a core test directory, and that means that for those particular genetic tests, every hub in the country has to be able to deliver those tests. So the same test is available for a patient no matter where they live. And there's also some specialised tests which are offered by 1, 2 or 3 laboratories in the country, so they can be referred for that particular test. So it's beginning to enable equitable access to genetic and genomic services around of England. It's limited to England. There's different things in place for Scotland, Northern Ireland and Wales. They have their own sort of restructuring of genomic services, but they don't come under this GLH structure.
Thanks, Jan. I mean, it's been fascinating for certainly Andy and myself to watch the journey of how genomics has changed in this last few years. I think, you know, we didn't realise when we were writing the curriculum for the Bioinformaticians back in 2012 that actually within about a year it would be out of date. And that's just shown how rapidly things changed. And it continues to evolve at that kind of pace, which brings its challenges as far as education, training, and all those sorts of things. So, John, what do you think, what are the key new services that are now being offered? You've talked about whole genome sequencing. So what other services have started to come through, and how has this impacted on the development of bioinformatics pipelines? So that's the way we analyse the genomic data and the way in which our bioinformaticians work.
So there's lots of new tests, and there's lots of consolidation, sort of bringing together of tests that were previously different. So people used to be offered or have a series of single gene tests. So you'd test one gene, was that the one with the mutation? No, right, we'll test another one, we'll test another one. So there was this serial sort of testing strategy, and I think it's a diagnostic odyssey where you sort of have test after test after test. So it's bringing those individual tests together in larger panels, which is a real sea change of the way of working for the laboratory side, for the wet lab side, as well as developing new algorithms and new ways to analyze those data. And also there's whole new services and there's very specialized services which previously weren't able to be offered, such as circulating tumour DNA testing, which is very novel. There's a lot of more of the RNA analyses which are coming online and will be sort of looking for particular gene fusions. So they're not single small nucleotide variants, which are quite easy to detect, but they're after translation, they're bits of the RNA that are stuck together incorrectly to make sort of protein combinations. So that's also something new. And there's services such as the rapid prenatal exome service, which is a highly specialized, quick turnaround service that's run from Exeter in the southwest GLH, which is available for the whole of the country for referrals. And there's also a huge sea change in cancer analysis as well. So cancer testing, particularly for solid tumours, has been quite limited to known what they call hotspot mutations that are well understood, well characterized variants that are pertinent to that disease. But with whole genome sequencing and larger panels, you can now look for a range of different variants that might have what we call driver status for that cancer. So are those mutations driving that disease, or are they just passengers? So that opens up a whole new opportunity as well. And it's not just new tests, it's what we call repatriation of tests. So like I said in your previous question, genetic labs previously had different specialities, and now each lab has to deliver a core set of services. So where you would previously send things away, you've now suddenly got to be able to deliver that in-house. So that's another whole set of learning that we've got to do. And the impact of that, particularly on bioinformatics, which is relevant to this discussion, is our programme of work we've had to undertake a real sea change in how we approach this. We've got to have scalable programming. We have to have reusable code because things need to be developed quickly. We need to deliver quickly and be able to validate and verify and have robust algorithms and processes for getting the data from the equipment to hand it to a genomic scientist for analysis. It needs to be flexible. We need to look at the infrastructure that we're using. That's got to scale up to be able to have sort of production level sequencing. It's gone from a single bioinformatician with their own sort of Linux virtual machine being able to support the entire laboratory. So we need now teams of people. We need to be looking outside of standard computing to look at high performance computing, cloud-based software as a service. It's the whole sea change. Automation and transparency are key. But it's not just the pipelines of getting the data out there. We need to look at end-to-end involvement, because bioinformatics now, it's an integral part of that laboratory test. The wet side, the sample prep side, and the instrumentation is only one half of that. The other half of the test is the bioinformatics. And so we need to apply the same rigorous laboratory standards to that bioinformatics part as we do to sort of the wet lab. So any sort of standards and protocols and checks you have in place for wet lab work, you also need to have for the data processing side. And there's an opportunity to get involved in more than just pipeline and data churning and processing and handing a result over. It's looking at data integration from different sources, looking at laboratory automation and things like that. So there's a lot of different opportunities.
Thanks, Jan. I think that's, yeah, there's so much there, and you've surmised actually what we've seen over the years coming through in some of the bioinformatic projects. You know, we went from looking at single nucleotide variants, we then had a lot of projects around looking at databases for variants. We then moved on to looking at building CNVs into our pipeline and looking for different kinds of variants. And then we see a lot more now, which is much more around the data validation, data standards, even exploratory projects looking at cloud integration and things like that. So I think things have changed, and actually the remit of the role has changed also massively over that time.
Absolutely.
And you sort of alongside both of us have watched the bioinformatics workforce grow since we started to train the bioinformaticians for the NHS back in 2013, and you now lead the doctoral level program, so-called the HSST, for bioinformaticians that it's integral with the workplace training. What do you think, what have you observed to be the main opportunities and challenges for them?
I think it's very much along the lines that Jan was talking about. So if you think about the sort of the axes in which a good clinical bioinformatician needs to work, sort of they need to understand the genomics aspects, which is one of the fastest moving areas of science at the moment, is incredibly driven by all the new techniques and methodologies. They've got to understand the informatics and the data science areas. Well, what's one of the biggest, fastest growing areas at the moment? It's all around big data, health data science, AI. Well, yeah, they need that. But then they have to make it work in a complicated environment. So the third axis, which matters hugely, is sort of the leadership into an organization that a bioinformatician can bring. So I think what we've seen has been this sort of transition from at the start, which was maybe not quite sure what the bioinformatician was for. And I think in the early days, there was a little bit of worry about it was about maybe fixing a printer. I think it is many, many years since we've heard anybody discuss it at that sort of level now. I think there's a real realization now that as medicine becomes much more digital, much more driven by big data, the bioinformaticians are at the forefront of that. And I'd say, I think what's exciting is it's the skill sets that people are developing in this area is actually applicable in so many other areas as well. So I think it's a really rich skill set that people have to develop. So really being a good clinical bioinformatician is hard because you have to understand the IT, you have to understand the biology, and even more difficult, you have to understand how you make that work in a complicated organization. And I think the changes we've seen has been just the development and the professionalization of the community involved in bioinformatics now, and the way people are getting their voices heard much more around being more in control of some of the developments that are now occurring in the NHS. So I'd say in terms of what it means to be a bioinformatician, I think it's becoming much clear now that actually to be a really good bioinformatician, it's not just, it's almost like too much to be an expert in all of it. You really do have to be someone who understands team science, enough of the different areas, and then actually be able to play a role in almost coordinating and pulling a lot of this together, and what are the right sort of teams you need to build. So I think it's becoming less of a sole bioinformatician and much more of a coordinated role. And I think what's been really exciting for us, I think, to see is the way that the community of bioinformaticians we've been involved in training has actually been able to work together, because you don't even have to be in the same trust to be able to work together. We're now seeing projects coming through, which is bioinformaticians across different trusts coming together to actually develop resources and tools that are useful across the patch. So Berkeley, I do not see the pace of change in this area slowing down at all in the next 5 to 10 years. So the problem we've had that the sort of syllabus needs to change pretty much every year, I think that's going to continue for a while longer yet. So it's just a really, if you're interested in being at sort of the nexus of about 3 really quite exciting areas, it's
I think you're right, Andy. I think, you know, what we've seen actually as well is it go from sort of 1 or 2 bioinformaticians in each hospital to now building teams, and teams that take on different roles, responsibilities. And I think actually picking up on your point around leadership, I think many of our bioinformaticians have been propelled into those sort of leadership roles really, really quickly. And I think it's something maybe Jan will come on to next, but and actually what has been nice, although I think it's, you know, it's been challenging at times, is developing those guidelines, developing those sort of that professionalism of clinical bioinformaticians that matches all the other wet lab side of the genomics landscape. Jan, so where do you see the greatest opportunities for our bioinformaticians working in the UK, and how do you think we really should support them to develop the right skills for this landscape beyond 2021?
I think Andy sort of listed quite a lot of that when he answered the previous question. And just to pick up on the leadership, it is important to recognise that in the mandated management structure that NHS England put on the GLHs was there had to be a lead bioinformatics scientist, which I think speaks volumes about where the profession has come. And it's on a par with having a lead for the rare disease and a lead for the informatics. There's also a lead for the bioinformatician at quite a senior level. So that sort of shows that it's being recognised more, that it's an integral part of the laboratory. In terms of developing the right skills, I mean, one thing that needs to develop, as long, you know, the technical skills, the platforms will change, they'll develop. So I think one thing is, and I don't know how easy it is to teach flexibility and teach people to be resilient and teach people to be aware of how to keep on top of changes. There's certain key skills that are needed, and I think the focus from the industry side or from the NHS side, we're very much focused at the moment on operations. You know, we need to get this test directory delivered. We need those hard technical skills. You know, we're under a huge amount of pressure to do a lot in a short time. So we need programmers, we need people that understand how APIs work. But you know, we're gathering all this data. So the other thing we need, like Andy said, is people that know how to interpret that, how they can mine it, how it's going to contribute to patients for the future. Because a lot of the cancer testing, although they're doing these big, large panels, there's only limited genes that actually make a difference to a treatment or make a difference to a diagnosis at the moment. But if we're sequencing 500 genes at a time, how are we going to make sense of that to improve future cancer patients' lives? And that's where the big data, the machine learning, those sorts of qualities will come in, because the data is just now starting to be gathered in earnest. So that's where the focus can be on the future once the operational side matures in the next couple of years.
Thanks, Jan. I think there's lots of food for thought for us here, and I think we could probably spill into another webinar on this sort of topic. But Andy, what would your advice be for anyone wanting to move into bioinformatics, and particularly maybe with a sort of clinical or translational sort of focus?
Right, so I say the very most thing I think is absolutely critically important is to remember that this is for a patient. So what you're doing in clinical informatics is to make something better for a patient. And if you're not doing it to make it better for a patient, then essentially, why are you doing it? So there's a very different ethos to being a clinical bioinformatician to being a research bioinformatician. You can't chase something down because it's sort of amusing and it might give a paper or something like that. You've got to be thinking all the time about what it means to a patient. So if you're moving into clinical bioinformatics, it has to be, and you really have to take on board the fact that there are patients, there are families, there's that whole interaction. And I think it's maybe something we don't think very much about because we're a bit behind the scenes, but always being up front and center that it has to benefit people at the end of the day. That's why you should, maybe why you should be going into it. And then it's also that you're going to be working in a really complicated environment. So a patient pathway is a big and complex thing. So that you can't, if you're going to be serious about going into clinical advice, into the sort of the clinical area, there is an understanding about how things actually work in a hospital setting, how a particular disease pathway works and what the various sources of information are. So really understanding where in that sort of patient pathway, that patient journey, the sort of the bioinformatics sits. So although to us it's big and exciting and it's huge, actually in terms of the context of the whole pathway, it's just one small chunk of what is a much bigger and more complex system. So I think to be a clinical bioinformatician, yes, you need the technical skills, yes, you need the bioinformatics, but you also need that bigger view of where it fits in to actually how it's going to support and help a patient. So I think if you want to move into that clinical bioinformatics, that translational bioinformatics, I think it's also taking on board how you get that experience and understanding of what you can do that will make a difference to a patient, not what you can do that will make in addition to knowledge. And I think that's, and I think we've all had experience now about actually that last bit about how you actually make it work in practice is sometimes a lot harder than you might think.
Thanks, Andy. Yeah, I think I guess just to pull these sorts together now on sort of that really picking up on some of the points you both said there and relating it to obviously we deliver a course which is a fully online course in bioinformatics, and whether it's that course or whether it's the course where we train the bioinformaticians for the NHS, there are a few key things, but in particular in relation to the online course, I think the point around resilience is key. So actually, we can teach people to program, we could teach people certain bits around NGS pipelines, but the key things are these are always going to change. Things are going to need adapting. We might be looking at cloud infrastructures. We might need to make a change to implement an ad looking at copy number variants, and it will need revalidating. So the key thing that we will do is teach you how to do some of these things, but in real world examples. So in a sense, we're teaching some of those skills around resilience, because we can never say specifically what you might encounter in a clinical workplace, but what we can do is take you through examples with a real clinical problem-based focus. And that's actually what we do. So we have a programming unit that's focused around working in teams, working in an agile way to develop actually a real clinical piece of software. We work on things like, working with APIs, for example. And we also mimicking that idea of working in teams as well. So, in a bioinformatics team, there might be different focuses on different aspects that need to be delivered, but those teams will also interact with other clinical scientists, other clinicians. You'll be looking at things like multidisciplinary teams. So getting you to work with your peers on the course that might come from very different backgrounds, whether it's sometimes computing, sometimes much more biological, more clinical, will actually get you to work in very interprofessional environments when we set you on those clinical problems that we use within the course. And importantly, one of the things we always link to are the way that the community, the genomics community, is developing clinical guidelines. So we always link back to what is the current status quo, what are the guidelines that are being used in the clinical environment. So I think that's probably all we wanted to say in terms of linking in with some of the things around skills, what the course can provide in this sort of space, but also actually what's happening on the ground right now. And I think both Jan and Andy have given a really good summary of where things are, but also where we need to go into the future. So I guess, Jess, I'm not sure what's cropped up on the questions, but I think at this point, myself and the panel will be very happy to answer any questions that anybody has.
Yeah, so there's been 1 or 2 questions that have come in. The most significant one that you might be able to look into answering is about a bioinformatician in the NHS and their critical analysis with regards to integrating into their pipeline. So they were specifically looking about tools that capture de novo mutations. Is this something you'd like to offer a comment on, any of you?
Yeah, I'm just having a read of the question. How does the bioinformatician? So there are guidelines. We adhere to national guidelines for validation and verification of code and tools. So what we're trying to promote is code review, first of all, so making sure that your code is right, getting someone else to check it. If your team's big enough, it's good to do pair programming to sort of co-develop it. In terms of validation, there are 2 different ways that we need to demonstrate. You can use a dry validation, so just running data through that tool. Does it get the results that you're expecting from the input data that you put in? And then you need to integrate that tool into your entire pipeline. And again, you can do what we call a dry validation, which is put samples through that whole pipeline with mutations you know should be present or variants you know should be there, and then check, do you get them at the end? What's your sensitivity? What's your specificity? In terms of de novo mutations, that's slightly more difficult because you're obviously looking for things that you haven't seen before. In the case of sort of rare disease or high frequency mutations, the gold standard is to check that those mutations are there by using a different method, and that method is usually Sanger sequencing for small variants, or in either single nucleotide variants or small indels. You can do Sanger sequencing to confirm that you actually found a real variant. That's slightly more difficult for fusions, but then we need to look at if there are any alternative technologies. If there aren't, it's more difficult, but you can assume a certain level of confidence if you can detect using known mutations. You've got a good sensitivity and specificity, and I think there are certain numbers around the actual numbers of samples and the actual numbers of variants that you've got to detect in the guidelines, and I can't remember those off the top of my head. And verification, after you've done a validation, a verification is just ensuring that that tool or that piece of software works either in your own hands or somebody else's hands in the clinical service. So it's not just good enough to say, oh, I've run 30 samples through this, it works. It's also got to work in real time for every patient on the computer that you choose to use it on. So that's your verification that it works.
Yeah, and just quickly to add in there, that's also where some of the new software engineering methodologies come in, because when you go to things like virtualization and containerization, you can talk about adding a new tool in and then running it parallel with what you've already got to see are they concurrent or not. So you might be bringing something new in, but with some of the new software engineering environments, it's actually easier to do that sort of continuous development and integration than it used to be. So that's also where you need to know not just about the biology of how you do the variants, but also then when you implement it, what are effective ways of implementing it so you don't mess up the existing service, because you're not going to make yourself popular if you bring a service down and you've got lots of patients to do.
I think there was a couple of questions around maths background and computer science background, so perhaps I'll just answer off those. So you don't particularly need a maths or a computer science background for our course. We take people from a biological science, clinical, medical background as well. so that's not essential. If there's areas you wanted to brush up on, there are lots of good MOOCs courses that we could recommend just to get some basic skills in, whether it's R or Python. Typically, most of our bioinformaticians tend to program in Python, but other people are doing bits and pieces on the more data science in R sometimes. So, but definitely a bit of programming background in Python is probably not a bad idea. There's a question as well, Jan, that I think you might be better versed to answer, which is to do with, are the clinical trial results in relation to cancer available in public databases, and what are the most useful data sources for cancer genomics in the UK? They're already aware of COSMIC and the Atlas Genome Project.
Yeah. So they're the main ones. There's also, but it's difficult. There's to actually get clinical trial information on the actual trials, then the CRUK has a national trials database, so you can look to what trials. In terms of molecular results, there is, I can't think of the organisation off the top of my head. They do collate molecular results from all clinical trials, but very often, if those clinical trials have a commercial element, then the data is protected for a certain number of years to allow the analysis and the publication to take place, and then they'll be made available. We do tend to put things in Geo. So that's a very large database, and that's sort of the standard. So if you have an academic publication out of the clinical trial, then the data will be available in the Geo database.
Thanks, Jan. I think we've got a question from Anna, and I remember Anna. Anna did her MSc in genomic medicine with us, but so this is around, how do we expect the transition from short read to long read sequencing in the clinic in forthcoming years? I'm going to hand over to Jan in a sec. What I do know is we're supporting more and more of our bioinformaticians to do metagenomic projects, so looking at particularly microbial sequencing using long read technologies. But Jan, do you want to comment what's been done in Manchester so far?
Long read sequencing is still very much in the research arena presently. We know that it's a gap. We know that there are limitations for short read sequencing. That technology is not available in routine practice at the moment. It would be good if it could. It would answer a lot of questions that can't be answered with short read chemistry. I think we're gonna see more and more of it. I'm attending a workshop next week, which is sort of an exploratory workshop with NHS England and a couple of instrument manufacturers to see sort of where steps could be taken to look at that translational work. It will become important, but it's not there yet.
And we're seeing more of the projects in that space are sort of fitting more into the infectious disease areas. Yeah, so if you think about the excitement that people might be having this virus and other things at the moment, you're seeing those are the sort of places that they're coming in. But yeah, it's this transition between it, to be able to move it to clinical, it has to be very safe and very proven if you're actually going to put it into the front line. So I think it's one of these ones it's moving its way down that translational pipeline. So it's certainly things we prepare people for, but it's not yet in huge use.
Thanks both. I can't see any more questions. Jess, is there anything that we've missed?
So if you just click into the Q&A box, there are a few.
Okay.
One of our attendees has also asked about whether this course would be applicable to the NHS system in Scotland. I'm not sure, Jan, if that's something you wanted to comment on. You mentioned that before.
I think it would. I think the issues are the same. I think that the difference in the devolved nations is not the technology or the tests, it's more of the process and the money and sort of the actual delivering that's different. I think the same technical, scientific and clinical questions are there in the devolved nations, but I think it's the setup that's different, not the actual science and the clinical need. So yes, I think that the course content would be absolutely relevant there.
I think there's also, sorry, Jen.
I was just going to just touch on another question about specific guidelines, and we're just we really follow the ACGS guidelines where they are available, and they're also influenced by some of the American guidelines. So that's just to answer that question quickly. ACGS.
Yeah, and that's definitely what we use in the course as well. ACGS, or which is strongly based on the ACMG guidelines, aren't they, in the main? In fact, we've just been updating the materials to cover the ACMG guidelines on CNV that came out, I think it was last year. If the workplace does not have high throughput automated genetic testing, is it beneficial to take the course? I guess it's probably better, Jean-Baptiste, to pick up with that maybe offline, because it really depends on where you want to go and what you want to do, because a lot of what we'll focus on, it'll give you good programming skills. There is some other, we focus a lot on the clinical genomics, the genomic background, but also you'll get experience in building a pipeline, but that's only sort of one aspect of the course. So there is lots of other stuff that we do on the other units. I suggest that get in touch with us and we can have a more in-depth conversation around that one. I think I'll probably wrap up at that point though, Jess. I think we've answered most questions.
Sorry, is it just asking Arjun? This was a course.
Sorry, I'm struggling because the sun's just hitting my screen, so I'm now struggling to see. Gone, Arjun, yeah. What's the next step for someone who's completed the course and wants to join the bioinformatics team on one of the hubs? Jan. So we don't, I suppose the important differentiation is to say that to become a clinical bioinformatician in the NHS, you become a registered clinical scientist, as Jan is, and this course can't actually clinically register you with the Health Care Professions Council, but it will give you a nice stepping stone.
I can quickly answer that this course will provide you with the fundamentals, and the step after that is there's a national training programme. There's the STP, Scientific Training Programme, which is run by NHS England. Sorry, the National School, funded by NHS England, and that's a 3-year training programme, and each hub puts in applications for places every year. Say we want 2 trainees this year, and that's a national competitive programme. The other way in, which is slightly more difficult, but if you have a lot of experience, it's worth looking at the Academy for Healthcare Sciences website and look at equivalents, because there's a different route through. If you can provide good enough evidence to show that you've been working in that area and that you have the same set of skills, you can put a portfolio together and apply for equivalence to that register. So if you've been working in laboratories and you have built up a few years of experience, it's worth exploring the equivalence route as well. So the National School STP programme, or look at the Academy for Healthcare Sciences website for equivalence.
Thanks, Jan. Great stuff. I think we've covered off.
Yeah, as you said, Ange, I think this could go on to many other webinars.
Yeah. I think, Jess, do you want to say about your contact details just before we wrap up?
So I'm just popping my email address in the chat box. You will all receive a follow-up email with the recording after this webinar, and also invitations about our future webinars, which we have coming up. We'll be offering a webinar about units 3 and 4 over the coming month and 2 months. So keep an eye out for those in the future. And yeah, we hope to hear from you via email with good questions, and we can always pass them on to our academic team for further information. But for now, thank you very much everyone for joining us.
Thank you, Jan, and thank you, Andy, as well, for joining me. Okay. Bye.
Bye.